All right, good evening, everybody, and welcome to the Jeffrey B. Graham Perspectives on Ocean Science Lecture Series. My name is Harry Helling. I'm the Executive Director here at the Birch Aquarium at Scripps. And it is my great pleasure this evening to introduce our speaker for this evening, Dr. Paul Jensen. Paul is a professor in the Marine Biology Research Division at Scripps Institution of Oceanography, University of California, San Diego. He obtained his bachelor's degree from Florida Tech, his master's degree from San Diego State University, and his PhD from the University of California, San Diego. Paul currently serves in a leadership role as section head for biological research at our institution. He has nearly 200 peer-reviewed publications, 12 patents, and nearly 20 more provisional and pending patents. Paul's research group is interested in microbes in the ocean. They look at the natural products or compounds that they produce, why they make them, and how to better exploit them for useful purposes. This includes addressing basic questions related to species, biogeography, and chemical ecology while crossing over into applied research in the area of drug discovery. Please join me in welcoming Paul for this talk entitled Marine Natural Products from Sea to Pharmacy.
Okay, thank you, Harry, and thanks everyone for coming. It's certainly a pleasure to be here tonight. And so just for fun, how many people here know that Scripps is part of UCSD? Okay, that's good. That's very important for our chancellor, for everyone to know that. How many people think Scripps was here first? Okay, that's important for those of us at Scripps that you know that, because yes, we were here first, and we're very proud to be members of the University of California, San Diego. And so, as Harry said, we're a microbiology lab, and I like to say that we work at the interface of marine microbiology and natural products chemistry. And so we particularly are interested in bacteria in the ocean that make these things that I'm calling natural products, and we're going to spend a lot of time talking about what those are tonight. And so we're interested in which bacteria in the ocean make these molecules, and so that raises fundamental questions in biology about things like species concept. How do you know who they are, and which ones are related to which other ones? We're interested in where they live, and that addresses the question of biogeography, and if we travel around the world, do we find the same microbes making the same molecules, or do we find different ones making different molecules? We're interested in why they make them, and that relates to the field of chemical ecology, and that's something that we really don't know nearly enough about, but it's a very important part of this field. And we're interested in how to exploit them for useful purposes, as Harry mentioned, and in particular, drug discovery. And so it was suggested that I personalize this to some extent. And so I contacted my sister a few days ago and said, I think there was this picture of me like pointing out with my father's mask and fins on. And I said, I think somehow way back when I knew that I wanted to have a career in marine science, although I grew up in New York City and didn't really have many opportunities to experience these things, but somehow I feel very fortunate that I was able to make a career out of doing something that I really enjoy doing, and that is marine science, and in particular this field of marine natural products where I've spent most of my career working. Okay, so when we think about communicating, we generally think about doing that verbally, right? And if you think about communicating in the ocean, you might say, well, yeah, I know some animals that make noises and maybe communicate verbally. But in fact, the real language of the ocean is a chemical language. It's not a verbal language. And if we think about chemistry, we can hearken back to the periodic table of elements, and these are all the elements that we know that occur on the planet. But the chemistry that's really the basis of this language is really carbon-based chemistry, okay? And so the field of science that studies this carbon-based chemistry we know as organic chemistry. Okay, next question. How many people was organic chemistry their favorite class? Yeah, no, there's a few weird ones out there, but yeah, most of us suffered through organic chemistry, right? Me too, and I'm really surprised that I spent so much of my career so close to organic chemistry. But nonetheless, I've learned to love it, and it's a great field of science. And what this slide just shows you is the diversity of organic molecules. And, you know, carbon atoms can form carbon-carbon bonds. This shows you that some of them could be single bonds, some could be double bonds, some could be triple bonds. They can bond to other atoms like nitrogen and oxygen and sulfur. We call these heteroatoms when we're thinking about organic chemistry. And there are a lot of different functional groups that are shown here, but the important part is that this diversity is what makes this language of the ocean with which organisms communicate with. But we're not just talking about any organic chemistry, we're talking about biochemistry, because a chemist can synthesize an organic molecule in the lab. So we're not so interested in those. We're interested in biochemistry, which is really the organic chemistry that occurs within living organisms, okay? And so if we go back and think about biochemistry, again, lots of sleepless nights trying to learn these things for exams in biochemistry. Okay, so biochemistry, okay, so when we think about graphics like this, basically what it's describing is when we take in complex organic molecules like carbohydrates and break them down for energy. In the process of doing that, there's a whole series of intermediates, which are all, of course, organic molecules. And there are these cycles that we know. And these cycles are similar across all life forms. And this is really what we would call primary metabolism, okay? So all life forms do these processes in a very similar way, and they're fundamental to life as we know it. But the language of the ocean that I'm talking about is not primary metabolism. It's what we call secondary metabolism, okay? And secondary metabolism is, in effect, this field of natural products chemistry, okay? So living organisms can make molecules outside of primary metabolism that we call secondary metabolites or natural products. And these molecules are basically built by stealing these intermediates from primary metabolism and incorporating them into much more complex structures. And so this graphic, which again is you can't see very well and it's very complex, but basically it's just telling us that what nature has evolved to do is steal these small molecules from primary metabolism, these intermediates, and build them into very complex structures using some very sophisticated enzymatic machinery. And it's this diversity of chemistry that really has been nature's pharmacopoeia, okay? So historically, this is what we've gone to in our search for drug-like molecules from nature. Okay, so we're interested in marine natural products, right? And so these are secondary metabolites produced by marine organisms. And so these molecules can have a variety of functions. They can be ecological in nature, but generally, of course, they're ecological in nature. These can be for defense or things like nutrient acquisition. Settlement cues is an interesting one, okay? So everyone can think about what an oyster bed looks like, right? Lots of oysters living closely together. Well, when oysters reproduce, they don't just reproduce right next to each other. They produce larvae that go up into the plankton, right? And how do those larvae, when it's time to settle, know where the other oysters are, okay? Well, they respond to chemical cues released by the adults, and that's how they know where to settle, okay? So natural products play important ecological roles in things like settlement cues. Also, mate recognition is a really important one. And so this is pretty remarkable, okay? So crabs generally mate when the female is molting. And so if you take a stone and you put it in water with a female crab that's molting and then give it to a male crab, the male crab will immediately start to try to mate with that stone, okay? And so that's how you know there's a chemical cue that it's sensing. And this story is even more complex because crabs are also cannibalistic, and the female is in big trouble that the male not only wants to mate with her, but might want to eat her as well. And so the female also releases a chemical that suppresses the male's feeding instincts, in addition to letting it know that it's molting. So this is really one of my favorite examples, and this has to do with copepods, okay? And so copepods are very small crustaceans. They're shrimp-like organisms. They live out in the open ocean in the plankton. And when it's time to mate, they need to find each other. And so here's a little graphic of what happens when a male copepod comes in contact with a tract left by a female. And so what you're seeing there on the top is the female swimming, on the bottom is the male swimming, and when the male encounters the female tract, he takes off right after her, okay? And so here's another example also with a copepod, but this time we can see them in real life. And so what you're going to see, this line through the center here, this is a scent trail that's laced with these pheromones or chemicals produced by the female copepod. And what you're going to see is the male copepod, when he encounters that, first he goes the wrong direction, and then boom, he takes off right after her. So I don't know what happened after this field of view, but you can see how powerful these chemical cues are. And of course, defense is also a really important function of these molecules. And it's really defense where when these molecules have these functions, this is where we start to get into the kinds of molecules that we're interested in as potential drugs. Okay, so if we talk about chemical defense, you might say, well, okay, I can think of some examples of things that are chemically defended. And then you might say, well, I can also think of some maybe less obvious, but when things are brightly colored, it often means that they're producing something as a warning, right? Like, hey, stay away from me because I'm toxic. One of my favorite examples in the microbial world of chemical defenses come with the leafcutter ants, okay? So I think everyone knows about these ants. They can cut down leaves from large areas of forests, and they don't actually eat the leaves themselves, but they take them back to their fungal gardens, which they feed the leaves, and the fungi grow on the leaves, and then the ants eat the fungus, okay? But there's a problem because there's a parasitic fungus that can infect their nests. And how do the ants keep this parasitic fungus out from their fungal gardens? Well, if you look closely on the cuticles of these ants, you can see this powdery type material. And for a long time, people didn't know what this was. But then eventually, they realized it was a type of bacteria that was living on their surface. And it turns out that these bacteria produce one of these natural products or secondary metabolites, and it turns out it's a very potent antifungal agent that's selective for the parasite of their fungal gardens. Okay, so they have their own antibiotics that they carry around. They have their own antibiotic-producing bacteria that they carry around on their cuticles to keep their fungal gardens free of these parasites. But we're interested in marine chemical defense. And so when you look at a coral reef, you see all of this complexity and you say, wow, it's beautiful, but in fact, it's a very vicious world out there of eat or be eaten. And anything that you see out there has figured out a way to survive. And so we know that predation is really the major driver of what reefs look like. And so the big question is, how do organisms avoid predation on coral reefs? And so there can be some obvious answers to that. And so you might say, well, okay, we know that a sea urchin has spines, and that's a type of physical defense. We can go look at our beautiful leafy sea dragons on display here, and they're very well camouflaged. It takes you a while to find them in the tank. And there are some behavioral mechanisms that fish use to avoid predation. And then we see these soft, fleshy animals that are brightly colored, and they have no obvious structural defenses. And when you see that, you can pretty much guarantee that they have a chemical defense, and that's why they're not being eaten. And in fact, we know this because there's been a lot of really great work done where people have addressed these questions. In this slide, we're showing some assays that people do to test for chemical defenses. And so these are pieces of sponges that were collected and extracted, and the organic molecules or natural products that are present in those sponges are put into food pellets and then given to fish, either in an aquarium or out on the reef. And you can show that these extracted materials taste bad to the fish, and that when you separate those materials into their individual components, it's usually just one or two molecules in those extracts that make the sponges taste bad, okay? And so we've learned a lot about chemical defense through these types of experiments. And it also, again, happens in the microbial world. Shrimp tend to brood their embryos, and these embryos are susceptible to infection by a fungal pathogen, or at least the shrimp are susceptible to infection, but it's been noticed that the embryos are highly resistant. And when you look at those embryos, they're covered by essentially a monoculture of bacteria, and those bacteria produce an antifungal agent that defends the embryos against infection by fungi. Okay, so there's a lot of things going on in the natural world that are mediated by these organic molecules, and these defensive roles are, you can already think, these are obvious ways we can take advantage of an understanding of these ecological interactions to try to go out and discover new antibiotics and antifungal agents and other molecules that can have potential to treat disease. Okay, so that takes us to the idea of marine natural product drug discovery, okay? And so starting really in the early 1970s, people began going out and collecting marine organisms and asking what kinds of molecules do they make, and do these molecules have the potential to function as new drugs? Can they be used for this purpose? And people really have done extraordinary efforts searching high and low in the oceans, including using submarines to collect from some of the deepest parts of the ocean. We've had some programs, these are some collaborators of mine, where we were working in Fiji, where we had a program for many years, collecting marine invertebrates. The guy on the left side here was working at Bristol Myers Squibb when we had a collaboration with them through an NIH, a government-funded program, to look for new drugs from marine sources. And in general, the way these studies have worked, our researchers will collect different types of these soft-bodied animals and extract them and look to see if those extracts have any sort of properties that might have medicinal potential. And we could even call this the golden age of marine natural products, starting in the 1970s, because basically no one had ever looked at the plants or animals in the ocean to ask if they are making new molecules. And basically everything that you picked up had new chemistry in it. So there was this amazing period of people finding lots of interesting new structures and studying the biological properties of those molecules. And the results have been productive. And so today, there's a reasonable handful of molecules that are being used in the clinic now that came from marine sources. A couple of these are from sponges. This is from a sponge. This molecule is from a sponge. Both of these are to treat cancer. This is another anti-cancer compound. This comes from a sea squirt. Again, an animal that lacks structural defenses. This is another molecule used to treat cancer. This comes from a sea hare. Okay, so sea hares are mollusks. These are snails that have lost their shells, and the reason they've lost their shells is because they're chemically defended, right? And it's these molecules that defend them that have proven to be useful for the treatment of cancer. This one's pretty interesting because if you're thinking about what I've been telling you, this painkiller comes from the cone snail. But this cone snail, these snails still have shells. So why would an animal that has an obvious structural defense be producing one of these toxins? Well, it turns out that these cone snails are actually predatory, and they hunt fish. And they use a neurotoxin to paralyze their prey. Okay, so that's a slightly different function for these kinds of molecules. It's not for defense, it's for prey capture in this case. But it turns out that these molecules are highly effective for treating people who have chronic pain. Okay, so there's been some pretty nice success stories in terms of finding new drugs from marine sources. But you also might be thinking, wow, how do you go out and collect enough of these cone snails or sea hares to actually develop something into a drug? And there have been huge problems in this field in terms of supplying enough of these lead molecules. And you might be saying, well, why not just make them in the laboratory? Well, in some cases you can do that. In many cases, because these molecules are so complex, they can't be synthesized in a commercially viable way. And so there have been many molecules that have their development has been stopped because they can't be obtained in sufficient yields. So how do we deal with the supply problem? Well, this is where the microbiology comes in, okay? If we can culture microbes in the lab, we have a renewable resource, and so we can, in theory, produce all of the material that we need. And so we know that microbes from land have historically been an important source of medicines, antibiotics, penicillin. We know these stories, right? And so what about microbes in the ocean? And do they represent a poorly explored source of new medicines? And if so, which ones should we choose? So if we think about what types of microbes we want to culture from the ocean, the first thing we did was say, well, which ones on land tend to be producing a lot of these interesting molecules? And if we sort of summarize some data from summarizing where antibiotics have historically come from, about half of all of the antibiotics discovered come from this one group of bacteria, which we commonly call actinomycetes. And so we looked at this information a number of years ago and said, wow, why don't we ask if there are any actinomycetes in the ocean? And if there are they making anything unusual? And so there's lots of ocean to explore. Actinomycetes on land tend to live in soil. So we wanted to look in ocean sediments to ask if we could find these bacteria and ask what kinds of molecules they might be making, and if these bacteria were really different from the ones that we see on land. And so for years, we've been going out and collecting samples. We can do that by diving and collecting them by hand in shallow water, or using electric reels and small type grab samplers like this here, or we'll go out on the ships, including the script ships sometimes, and collect deeper sea samples in this way and ask questions about whether we can isolate these bacteria, and in fact we can. And so that was the first part of the question. The second part of the question is, are they different from the ones that we see on land? And we can use a variety of techniques. Anyone send a sample of their pet to understand what breed it is? Or has anyone done 23andMe to look at their ancestry? Okay, so we can do the same kind of thing for microbes as well. And we can figure out who they are by sequencing their DNA. And when we look at these actinomycetes that we're culturing from the ocean, we can recognize lots and lots of them, which are circled in red here, that are different from ones that have been seen on land. And so what I want to do is spend a little time talking about my favorite group of bacteria, which is the one with the red arrow pointing to it there. We found this group a long time ago and originally called it MAR1, and we subsequently gave it the name Selenospora. And we spent a lot of time studying these bacteria in my lab and with some of my collaborators. Okay, so here they are. This is an introduction to this genus that we've called Selenospora. They're very photogenic as far as bacteria goes. They're brightly pigmented. We've named 3 species to date. We're in the process of naming 6 more. And we really got interested in these because when we tried to grow them in the lab without seawater, they wouldn't grow. And that suggested to us that, one, no one had ever seen this before with this type of bacteria, but it also suggested that they were adapted to life in the ocean, and maybe that means these adaptations include the ability to produce some molecules that we've never seen before. So we've spent a lot of time collecting samples around the world and looking to see if we can find this group of bacteria. All of those colored circles represent places where either we've found them or other people have found them and reported them in the literature. And so we've colored those circles based on the 3 species, and so you can see that there's some patterns in terms of where the colors occur, and that gets to this question of biogeography and bacterial distributions. I'm not going to spend time talking about that today. You're probably noticing that there aren't any colored circles very far north or very far south, and you're probably wondering if that's just because we don't like to go to cold places, and we don't. But we have taken samples from up in Alaska and also from some deep sea sites, and we've been unable so far to culture these bacteria from any of those locations, nor have any of the other people that have been contributing to this data set. So for reasons that we don't understand but that we appreciate, these bacteria like to live in nice warm places. Okay, so the next question is, do they make any interesting molecules? Okay, and sure they do. And so I've been collaborating with an organic chemist at Scripps named Bill Fennicle, and he's one of the pioneers in this field of marine natural products chemistry. He was one of the first people to think about going out into the ocean and collecting seaweeds and invertebrates and other things and looking at the molecules that they make. And we've worked together for many years on this group of bacteria, and this is a representation of the molecules that these bacteria produce. And so once again, you can see enormous structural complexity, and so that's a good sign. We like to see that in terms of the hopes that some of these molecules may end up being useful as drugs. And so when we add up all the molecules that we found, more than 60% of them are new. And so I think that would argue if we go to new places and find new microbes, they're going to be a good source of new molecules that we can study. And so I'm going to highlight in red there a molecule that's really been the most important one that we found from this group. And this is a molecule that we call selenosporamide A. And so selenosporamide A is produced by one of the 3 species, Selenospora tropica, and it's a pretty small molecule by secondary metabolite standards, but it has this really unusual bicyclic ring system, which is highly reactive. Okay? And what we know about this molecule is that it's a very potent inhibitor of a structure in the cell called the proteasome. And all eukaryotic cells have proteasomes. They're basically the garbage disposal for proteins in the cell. And proteins are very important in regulating the cell cycle. And it turns out that when you inhibit the proteasome, you selectively kill cancer cells, because in cancer cells, they're growing out of control, and there's no way to inhibit the cell cycle. So by selectively inhibiting the proteasome, you can selectively kill cancer cells. And that's a proven mechanism that's been established with one other drug. And so this is the worst slide I'm going to show you. But basically, through some hard work by a lot of people, we know exactly how this molecule interacts with the proteasome and how it inhibits its function and how it does so irreversibly, which is what makes it such a very potent inhibitor of this cellular structure. What we've learned is relatively recently is that this molecule, probably because it's so small, that it can also cross the blood-brain barrier, okay? And so that's a big one. We all know that cancers like glioblastoma are very difficult to treat with chemotherapy. And so this molecule has been developed by a number of companies, and most recently it was picked up by Celgene, which is here in San Diego. And Celgene has taken this into phase 3 clinical trials, and so I think many of you know there are 3 phases to clinical trials, so this is the last phase. And if it proves successful, then we will see the first marine microbial natural product to become a useful drug. So we're very excited about that prospect, and so keep your eyes out. If it does work, we'll probably see something in the paper about this, because it'll be a pretty big deal. So we're excited that Celgene picked this up, and discoveries like this, they do a lot for the entire field, right? The National Institutes of Health invest money in this field because they want to see new drugs discovered, and so everyone's grant proposals that work in this field will be viewed more positively when there are examples of real drugs developed through this type of science. So we're very excited about that. So everything that I've told you so far in terms of microbial natural product drug discovery, I'll say was discovered using what we'll call a traditional discovery paradigm, okay? So we go out, we collect some sediments, we isolate strains of bacteria from those sediments, we culture them in the lab, we extract those cultures often with organic solvents, then we'll test those extracts in some sort of bioassay, like ask if they can kill cancer cells. And if they can, then we'll do some type of chromatography to separate those extracts into their individual components and try to isolate the molecules that are responsible for the activity and hope that they're new, okay? So that's how natural product drug discovery was done for many, many decades. But today, we can use new approaches. And so we all know about genome sequencing, and the ability to sequence DNA has transformed so many different disciplines, including ours in the marine natural products world. And so now we can do this process that we call genome mining. So we can isolate strains of bacteria, and instead of growing them and hoping that they make an interesting molecule, we can extract their DNA and sequence their genome, and we can ask if we can predict that they'll make an interesting molecule. And the first time we did this was we sequenced the genome of a strain of Selenospora tropica, and then we can go around and we can look throughout the genome and identify all the genes that we think are going to make these kinds of molecules that we're interested in. And we can even look in more detail at these genes, and believe it or not, you can read these almost like a book, and you can make predictions about what the structures of the molecules are going to look like from reading these gene sequences. And once we make these predictions, that can help us with the discovery process, because it allows us to find the molecules more easily. It also allows us to ask if these are the types of molecules structurally that we really want to go after. Okay, so this whole genome mining process has really made this field a lot more exciting and a lot more fun, because it's a lot more sophisticated now. We don't have to just hope we get the right microbe and hope that it makes a molecule of interest in the laboratory. We can now take a very informed approach and read its genome and make predictions about who's going to make what. And so there are bioinformatic tools that can help us do this. We can take our genome sequences. You used to have to do this manually, but now you can plug these genomes into these tools, and you can get a whole lot of information out that will tell you things like, well, this strain's going to make some molecules that we already know about, so let's not study them, whereas this other strain looks like it's going to make something new and interesting, so let's spend all of our time on that strain and try to find what those products are. So this concept of genome mining has really revolutionized the field of microbial natural products research. And so we can even take this to the environment. It doesn't even have to be based on an individual strain, okay? So we could actually go to an environment and probe to see if the types of genes that we think are most important are present or not. So instead of just collecting random samples and hoping that we isolate the right microbe, we can first ask which samples have the right microbes in them with the right genes before we even start that process, okay? So there's lots of really powerful things we can do now that we couldn't do before. And a lot of people are taking a synthetic biology approach to this. Okay, so when we grow bacteria in the lab, they don't necessarily make all the molecules that they have the potential to make. Okay, and so remember, I said that they're making these molecules by borrowing intermediates from primary metabolism, right? Primary metabolism is all about growth. Okay, and so if it's to your advantage to be able to grow and be healthy, you don't want to have to divert things that you need for growth to make these toxins if you don't need to, right? And so we think a lot of microbes don't make these molecules unless there's some pressure that they sense, and maybe something's trying to eat them, or maybe they're competing with other microbes. And so when we look at these genomes, a lot of the genes appear to be silent. Okay, they don't appear to be expressed in normal laboratory culture. And so people can use this field of synthetic biology to try to take the genes out and put them in another organism and make the molecules that way. In my lab, we're a lot more interested in what the natural cues are to make these molecules. We know microbes know how to make them. We just don't know what the conditions are to make them turn these processes on. And that's something that we're really interested in, and that relates more to this area of chemical ecology that I was talking about. And so we can see really amazing things now. As we sequence these genomes, we can start to even see how microbes evolve to make new chemistry. So once we know what the genes look like that make these molecules, we can see how they differ between related groups of bacteria. So here's our friend Selenosporamide A that I told you about that's in clinical trials. We can predict that the ability to make this molecule was acquired by these bacteria about here in their evolutionary history. And then as these two lineages diverged from each other, this species makes one version of the molecule and this species makes a different version of the molecule. And when we look at the gene clusters, we can see exactly which genes were lost in this species. And that explains why it doesn't have the same structural features as the other one. So this is really fascinating. So now not only can we find new molecules, but we can start to understand how they evolve in different organisms. And then we can start to ask why. Why does one species make one version of the molecule and one species make another version of the molecule? And so these are really important questions that we're addressing in our lab now. Okay, so what brought me here tonight was a really great program that's offered by the University of California. This is called their multi-campus research programs. And what the university has done is said, we have all these campuses and all these people doing interesting work, but they're not really talking to each other. And so why don't we offer people grant support if at least 3 different campuses who've never worked together before will come together with some kind of program? Okay, so myself and a couple others responded to this call, And what we proposed to them was that here at UCSD in my lab, we would do some of this genomics and microbiology stuff that I've been telling you about. And I contacted a colleague at the Santa Cruz campus who's really good at the natural products chemistry part of this. And I said, well, we need another campus. And he said, well, I know some people at UC San Francisco who have this pretty amazing new technology they just developed for high throughput screening where they can predict the targets and the functions of natural products. And so we got together, we put a little proposal together, and we were fortunate that it was funded, and part of many grants now have outreach components to them where the funding agency wants us to come and talk to people like you to tell you what we're doing. And so that's why I'm here today. And so I finally would like to acknowledge, we're just so lucky to be here at UC San Diego. I mean, the students that want to come here and study with us, I mean, they did a lot better in school than I did, I can tell you that. It's a pleasure to work with them. It's a pleasure to come to work. Not only do we have a great view, but we have a lot of great colleagues, and we really have a nice time doing research here. But if there's time, I'd be happy to answer questions.
Do you identify the atoms first, one at a time, or how do you put the molecule together?
Yeah, there's various techniques to solve the structure. This is a very specialized field, and so there are two primary techniques that we use. One is called mass spectrometry, and basically that tells you the mass of the whole molecule and in some cases the molecular formula. But the technique that's really most critical is called nuclear magnetic resonance spectroscopy, NMR, and that allows you to see the atoms and their relationships to each other, and that is how organic chemists solve these puzzles, by looking at NMR spectra.
You never mentioned the atom at all in this speech. You don't mention that word, atom, at all.
No, I didn't. Yes, so no, I didn't mention the word atoms. You're correct. After the periodic table, we were done with elements and moving on to molecules.
I think you mentioned you were harvesting maybe soft corals, was it, in Fiji? Was that the strain of the S. tropica that you referenced? What kind of soft corals was that found in?
It actually came from a sediment. And so although we have seen them in association with plants and sponges and other invertebrates, and so the original strain actually came from the Bahamas, although we found them in Fiji as well. And we think because they live in ocean sediments, and sediments often get disturbed, that especially filter feeders like sponges will pick up the cells, and when we collect a sponge, we can see them there. But we think their primary habitat is in ocean sediments.
I was curious, is there a name yet for the Celgene product that's in its clinical trials, phase 3? And when, is there a prediction as to when it might be released or put on the market?
Yeah, so you can go on Celgene's website, and they list all of their molecules that are in clinical trials, and you'll see this one as having entered phase 3. And so I like to call it the name that we gave it, which is slinosporamide A, but it's had about 4 names since then, because the companies that develop it always give it other names, and this is now going under the name of marizumab. Yeah, so you'll find it there on their website, and they do have some predictions. I think there's about the phase 3 trials actually take quite a while, and I think there's going to be at least a year and a half before there's any word on the results of those trials.
Hi, could you speculate or give any results on why a marine bacteria would have something that's so powerful against a eukaryotic cell?
Yeah, thank you for that question. And we think about that all the time. And so, some of the molecules they produce are antibiotics, and that kind of makes sense because there are a lot of other bacteria living in ocean sediments, and it would make sense that they're using antibiotics to compete. But why would they make something that's not going to affect other bacteria, that's going to target eukaryotes? And so I didn't really go into much detail about the life cycle of these bacteria, but they tend to grow very slowly, and they produce spores, and they're non-motile, okay? And so you could imagine a spore that's sitting in ocean sediments, and maybe there's a plankton bloom and a nutrient pulse, and that spore germinates, and they actually grow as filaments and form what we call a mycelium. And when the nutrients start running out, they produce more spores, and those spores get distributed again, okay? And so when they're growing as a little clump, that's a little ball of nutrition for something that likes to eat bacteria in ocean sediments, which there are a lot of invertebrates that do that. And so our working hypothesis is that this molecule is produced as a feeding deterrent. And so just like I talked about soft-bodied invertebrates producing feeding deterrents, so fish and other things don't eat them, we think bacteria have those same kinds of pressures in ocean sediments, and these bacteria in particular. So we've been doing some tests in the lab to try to gain support for that idea.
So how easy is it to work with the Selenospora? So you said it grew slow. Do you clone, or how do you sort of?
Yeah, they grow slowly compared to things like E. coli, but we can culture them in the lab without a problem. They come up pretty slowly on our plates when we try to isolate them, but if we use the right selective methods, we can see them. And so it hasn't really been a problem to grow them in the lab for these discovery type studies. I should point out that a lot of bacteria are problematic in that sense, but these aren't. And what I'm really happy about is that the material that's being produced for all these clinical trials is actually being produced by fermentation in the natural organism. Okay, so many times people will tinker with the organism and engineer it, or as I mentioned, they might take the genes out and try to put it into another organism that might make more of it. But here we actually have the native strain that's producing the molecule that's being developed through clinical trials.
You said that, it's supposed to fight cancer. How does this molecule actually just attack the cancer and not healthy cells if it attacks eukaryotic cells?
Yeah, well, this is a problem with really all chemotherapies, and they're not only toxic to the cancer cells, but they're also toxic to normal cells. And so chemotherapies rely on the fact that cancer cells are growing much more rapidly and tend to take drugs up more quickly and are more susceptible to them than normal healthy cells. And so this is where dosage becomes very important, right? And this is why even with the right dose, there are still usually adverse effects associated with chemotherapy, because inevitably some healthy cells are going to suffer as well. But the important thing, I think, is, if you can show the right type of remission and regression of tumors, is, especially tumors like glioblastoma, which are so hard to access with chemotherapy, that there's a lot of utility there.
You had talked about about seawater bringing alive some of your molecules, and so much of the discoveries are within the tropical zone. What about finding some of these molecules and bringing them back to life, if you will, in ancient seabeds? Are there opportunities there, or are the molecules just too deteriorated?
Well, it's an interesting question, and we've actually thought about this. I tried to get you excited about the evolutionary aspects of understanding how these molecules evolve. And here at Scripps, we actually have a deep sea collection of cores. And I was talking with one of my students, and I said, wouldn't it be amazing if we could go into some of those cores? And of course, as you go down into the core, you're going back in time, right? And we said, what if we could go back in time, and since our bacteria are spore forming, what if some of the spores are preserved in these deeper sediments? And what if we could actually culture some of the ancestors of the bacteria that we culture today and ask what kinds of molecules they were making? And we tried that, and unfortunately it didn't work. But yeah, it would be pretty neat if we could do something like that.
Well, as a geologist, I love ending on a geology question. So I want to thank you very much, Paul. That was excellent.